Hello, my name is Ben Ebert. I'm going to give a talk today on MDS, genetics, and inflammation. I'm at the Dana-Farber Cancer Institute and Howard Hughes Medical Institute. These are my disclosures. As you know from previous talks today, MDS and leukemia arise from a sequential acquisition of somatic mutations that initiates from individual hematopoietic stem cells that acquire a somatic myeloid malignancy associated mutation that leads to a clonal expansion, which is called clonal hematopoiesis. One of those cells may get another mutation that leads to further expansion of that clone and ultimately the development of MDS and potentially progression to secondary AML. Now, I wanted to highlight another type of clonal hematopoiesis or different types of clonal hematopoiesis and how they can progress to both myeloid and lymphoid malignancies. The field of clonal hematopoiesis is focused to date largely on point mutations or small insertions and deletions in individual genes associated with myeloid malignancies. We know that that is very common, occurring in about 10% of everybody by the age of 70, with at least amongst those with large clones with a variant allele fraction of at least 2%, which is what we call CHIP. In addition, there's a somewhat separate field that's grown up of investigators studying mosaic copy number abnormalities. Those are larger chromosomal abnormalities, gains and losses of chromosomal material that are very large. And we know that that's associated with disease because we've known for decades that there are copy number changes in MDS. That's why we get cytogenetics in all of our patients. And we know that there are cytogenetic abnormalities in other hematologic malignancies, both lymphoid and myeloid. In addition, there is clonal hematopoiesis without known drivers. These are expansions of cells that can be determined by skewing of X inactivation or by the accumulation of passenger mutations at a similar variant allele fraction. That may in fact be the most common type of clonal hematopoiesis. It's more difficult to define, it's more difficult to detect clinically, and its significance is less clear. However, either copy number abnormalities or point mutations can be readily detected. Now, we've recently looked for not only myeloid mutations, but also lymphoid mutations, looking for mutations that we know to be recurrent drivers of lymphoid malignancies in clonal hematopoiesis. We looked in 50,000 cases from the UK Biobank, and we detected both myeloid and lymphoid mutations at significant frequency. Clearly, myeloid mutations are more common than lymphoid malignancy, lymphoid mutations. However, they are present, and they are both age associated. So myeloid CHIP or mutations in myeloid genes in clonal hematopoiesis is most common, but lymphoid CHIP is also present at a lower prevalence. And the consequences of these mutations is as one might expect. Individuals with mutations that we see in myeloid malignancy genes, seen on the left here, have an increased risk of developing a myeloid malignancy. The absolute risk remains low, only a few percent over the subsequent decade or so, but highly significant increased risk of myeloid malignancies. In contrast, those with lymphoid CHIP or lymphoid malignancy mutations, associated mutations, have an increased risk of lymphoid malignancies, but a minimal or no increased risk of myeloid malignancies. And that corresponds to a hazard ratio of what's been very consistent of between 5 and 10, a hazard ratio of between 5 and 10 for myeloid malignancies, as well as a hazard ratio of about 5 for lymphoid malignancies. Now, in addition, if we look at copy number abnormalities as determined by what's called mosaic copy number abnormalities, this is using SNP array data from about 500,000 individuals from the UK Biobank, we can see that there are myeloid malignancy associated MCAs, and they are powerfully associated with the development of myeloid malignancies. And many of these are very well known to us, including deletions in 5q, 7q, 20q, and having one of those copy number abnormalities is highly associated with the development of myeloid malignancy. In addition, we can detect copy number abnormalities that are associated with lymphoid malignancies, and these are powerfully associated with the development of lymphoid malignancies, in particular associated with the development of CLL and other circulating lymphoid malignancies. It does not appear that there's a powerful increased risk of nodal lymphoid malignancies like diffuse large B-cell lymphoma, in which the mutated lymphocytes may not be circulatory and we may not be able to detect those mutations from a blood draw. In addition, it's consistent with other studies of clonal hematopoiesis and MDS. Having multiple mutations is worse than having single mutations. In particular, having both a myeloid chip mutation, a myeloid point mutation, and a myeloid copy number abnormality is associated with a greater increased risk of a myeloid malignancy, about a 30-fold increased risk. And on the lymphoid side, similarly, a lymphoid point mutation plus a lymphoid copy number abnormality is associated with a hazard ratio of about 100 for the development of a lymphoid malignancy. So the more mutations or the combination of a mutation and a copy number abnormality is associated with a very dramatic increased risk. And I think this starts to have implications for our diagnostic boundaries between CHIP and MDS or lymphoid malignancies like CLL. In addition, we know that blood count abnormalities integrate with these molecular abnormalities to further increased risk. So individuals who have an abnormal CBC and have any type of myeloid genetic abnormality, either a point mutation or copy number abnormality, have a powerfully increased risk of developing a myeloid malignancy. So a cytopenia plus a mutation that we see in MDS or a copy number abnormality that we see in MDS is associated with a really dramatic increased risk of a myeloid malignancy. And very similarly, on the lymphoid side, lymphocytosis plus one of the molecular abnormalities that we see in CLL, such as a point mutation or a copy number abnormality, is associated with a nearly 1,000-fold increase risk in the development of a lymphoid malignancy, predominantly CLL or SLL. And this work in aggregate is currently in press at Nature Medicine. I wanted to change gears briefly now and talk about the associations between these genetic abnormalities, either in clonal hematopoiesis or in MDS, and the risk of non-malignant disorders. Both CHIP and MDS are associated with an increased risk of the development of cardiovascular disease, either atherosclerotic coronary heart disease or stroke. And this has been now replicated in many different cohorts by a number of investigators. And the directionality of this finding has been shown in mouse models where somatic mutations in TET2 or DNMT3A or JAK2 can lead to accelerated atherosclerosis. And these mutations are associated with an increased risk of or an increased expression of inflammatory cytokines. And in humans, as well as in mice, the previous slide was in a mouse model, the presence of CHIP is associated with higher expression of the same cytokines, including interleukin-1 beta, interleukin-6, and for some mutations, interleukin-18. Now, those findings have been published and replicated. I wanted to talk briefly about another common disease, which is COPD, chronic obstructive pulmonary disease, or emphysema. And there's been a few papers on this topic, one by Zink et al., which was a large study of about 11,000 genomes and exomes. And they had smoking status and looked at the diagnostic codes, the ICD-9 codes for COPD, and found an association between CHIP and COPD even after controlling for smoking. but not. They saw an association between CHIP and COPD, but it was not statistically significant after controlling for smoking. A much smaller study looked at 2,500 healthy individuals, did not see that association between smoking and CHIP. And there was an association between a combination of COPD and asthma and CHIP, but not significant when corrected for multiple hypotheses. And finally, a study using whole exome sequencing and SNP array data from the UK Biobank showed a strong association between smoking and CHIP. So to really extend these findings, we've looked at a very large set of cohorts encompassing over 50,000 or about 50,000 individuals, including a cohort called COPD Gene, which was assembled specifically for the study of COPD and had highly quantitative phenotyping of these cases for their pulmonary function, as well as exome sequencing data or genome sequencing data that we could examine for the presence of mutations. We found that in these data sets in aggregate, we were able to see an association between CHIP and COPD, as well as an association between CHIP and severe COPD. This association is most strong in the COPD gene cohort, which is rigorously phenotyped for COPD. In other cohorts where we're relying on ICD-9 codes, which are notoriously difficult or notoriously inaccurate for COPD, the associations are a little bit weaker, as one would expect, but the association is strongest in COPD gene and in a meta-analysis for all studies. In addition, we looked at quantitative assessment of COPD, the FEV1, which is a determination of the forced expiratory volume. And here too, we were taking away the exact diagnostic boundaries or diagnostic codes for COPD, but really looking at quantitative effects on pulmonary function. We again saw an association between CHIP and pulmonary function, and with CHIP associated with decreased FEV1 %, as one would expect for an association with COPD. Finally, we wanted to look at the association between CHIP and smoking, which is important for the risk of MDS as well. And we were able to look at actual pack years of smoking, since these COPD cohorts quantified the number of pack years per individual. And we did see a significant association between CHIP and smoking, but it is relatively modest. So the increased odds of CHIP and likely therefore MDS is about 0.3% per pack year. So a 10-year pack year history of smoking was associated with about a 3% increased risk of CHIP, and a 50 pack year history of smoking with about a 16% increased risk of CHIP. And finally, we were able to analyze in a mouse model that mice with TET2 mutant blood cells compared to mice with wild type blood cells, when exposed to smoke in a controlled experimental setting, had increased emphysema with changes in their alveoli and quantitative measurement of alveolar cord length after exposure to cigarette smoke. And you can see here that mice with TET2 knockout had a significantly worse airspace disease, increased alveolar cord length, and therefore worse emphysema. So I wanted to tell you here today about how CHIP is common. It's associated with age, increased risk of hematologic malignancy, including MDS and leukemia, increased overall mortality, increased risk of cardiovascular disease, and increased risk of COPD. CHIP mutations and mosaic copy number abnormalities independently increase the risk of hematologic malignancy. Myeloid clonal hematopoiesis, including both CHIP mutations and MCAs, are associated with myeloid malignancies. Lymphoid point mutations and copy number abnormalities are associated with CLL and other circulating lymphoid malignancies. And the presence of genetic alterations in a combination with CBC abnormalities amplify the risk of developing the respective hematologic malignancies. The work I described on clonal hematopoiesis with both lymphoid and myeloid abnormalities was led by Abhishek Narula, and that is in press at Nature Medicine. And the work on COPD was led by Peter Miller. Our work on clonal hematopoiesis has all been in collaboration with Sid Jaiswal, as well as Pradeep Natarajan, Alex Bick, and the COPD work with a group of COPD investigators at Brigham and Women's Hospital.